Hello, everyone. Thank you for joining us. My name is Daryl Urbanski, your host as always, and today we are joined by a very special guest, MD, MDMS, Robert W. Malone, inventor of the core mRNA and DNA vaccine technology. He's a worldwide expert in RNA technologies. He's the CEO of RW Malone MD, LLC, consultancy and analytics in the biosector. He's a C-suite coach, expert witness, also does commercial intelligence. He also builds and manages teams to solve complicated problems in this space. So in 1989, he published a paper titled, I'm going to butcher this, cationic liposome-mediated RNA transfection, and he went on to get over 9 patents in the field. So I've asked him to join us here today so we can talk about mRNA vaccines, the current state of the world, and hopefully shed some light on what's going on. So Robert, thanks so much for joining us, my friend. How are you doing?
I'm doing okay, and it's my pleasure, Daryl. Thank you for the opportunity to speak to you and also to chat with your listeners. I look forward to it.
So we're excited to have you here. Before we even get into this, I want to share something like this because there's a lot of misinformation going around. I want to talk about how there's a quality of information, quality of evidence pyramid that typically starts with expert opinion and works its way up through various forms of evidence, like peer-reviewed evidence and verifiable evidence. And so even before we get into this, because when I mentioned that I was going to be interviewing you, I got some people, kind of say a lot of different things. You're asking about this. So this is clearly his profile on Google Scholar. And we can just talk about, when we looked at the pyramid of evidence, the studies here, his depth of knowledge and experience in this field is, I don't want to say unparalleled, but it's obviously vast. I mean, there's the 9 patents alone. If we really want, I can go through and show all of these things. Like, again, I want to get this out of the way because even he expressed at the beginning of this call how it can be kind of tiresome because, the media spins things. And I actually want to talk about this a little bit later on in the call. Some of the propaganda machines, all that stuff, we can get into that later. But I just want to say, like, as far as someone who can speak to these topics, I really don't think you're going to sit bedside with anyone else who's more qualified at the moment. And that anytime you do speak to someone on any of these topics, that you want to make sure you're getting valid information. And so I'm not trying to, in this conversation, I don't think we're trying to decree anything to anyone. We're not trying to declare this. We're just kind of inviting you to do the research for yourself, that the information is there. And if you know who to talk to, who to ask, because everyone has an opinion. And Robert's got an opinion. And I'm trying to just vet that Robert is clearly very qualified to answer some of these. And I'm going to do my best as we talk to kind of back up some of the stuff we're talking about here on Zoom, whether we talk about what's on the screen or not. So that's really it. So now, Robert, there's been so much fanfare going on. Before we even get into any of that, how did you even get into this? Like, what's your origin story? How did you even get into mRNA data? And you were a physician at some point, weren't you?
I still am. I'm licensed to practice medicine in the state of Maryland. I maintained my license, just renewed it the other day.
Okay.
But I don't practice. I manage teams and I manage clinical trials and design clinical trials as part of my kind of core competency.
Mm-hmm.
But in terms of the origin story, that's a good 10 minutes.
Okay.
I'll try to make it brief.
Okay.
I was originally a computer science student at Santa Barbara Community College. I got a bunch of awards there. I was a straight A student, and I was recognized as the top computer science student. This is 1982. I like to make the joke that if I had stuck with that, I would have been way better off financially than I am now, having gone medical track. There was still some punch card work back then. And it was absolutely the early days of Tandy and my dad was an electronics engineer. He built a computer himself using a kit. So that's kind of where I started off, and it's where I learned my logic skills. I was actually being a coder for a couple of intensive years, really taught me a lot about how to think and how to structure logic. But I decided that my mom had always said that she wanted me to be a physician. And when I left the community college and went to UC Davis for my last 2 years of undergrad in biochemistry, And at that time, they didn't even have a molecular biology degree. So went to UC Davis for a pretty hardcore biochem bachelor's in science. And I really, really wanted to do bench research. And at the time in the 80s, early 80s, it was just wicked hard to get into medical school.
Okay.
And to think about getting into medical school was to be the ultimate egoist. And so I felt like I had to kind of hedge my bet, something your community will appreciate, that kind of logic. And so I figured I could, if nothing else, become a scientist, if not a physician.
It's a noble career.
I ended up being both in the end.
Okay.
But my mother was really worried about breast cancer, as were a lot of people, and for a good reason. And I wanted to do bench research as an undergraduate. So I looked for internship opportunities, and I was accepted into a lab in the Department of Pathology, School of Medicine at UC Davis, with a very senior pathologist that was focused on mouse models of breast cancer. And that lab was tightly affiliated with the Primate Research Center at UC Davis. Mm-hmm. I later became a Primate Research Center investigator. And while I was there getting, I was kind of like the lab pet. I worked my can off. I was all the time at the bench, doing advanced molecular biology, southern blots, northern blots, which required me to work up RNA from mouse samples and mouse blood, et cetera, and mouse tumors. And while I was there, this little problem broke out in San Francisco and certain urban centers of gay men developing an immunodeficiency syndrome. And one of my two mentors, Murray Gardner, who used to be he really was the founder of the Cancer Center at USC and had become chair of Department of Pathology. Murray, together with Preston Marks and others, recognized in 83, 84 that there was an immunodeficiency syndrome being passed around in the monkeys. And so they hunted that down with electron microscopy and identified the first retrovirus associated with immunodeficiency syndrome. That was published in Lancet, and then that kind of set off the subsequent cascade. Murray actually went with Bob Gallo to visit Luc Montagnier, I think it was in 84. That's immortalized in the film In the Band Played On, HBO. And he came back, literally came through customs with the virus that causes AIDS in his pocket. And I remember him coming into the building, just bouncing down the hall, talking about this, that he got this through customs in his pocket. And that set off a whole cascade that immersed all of the people around me that I was embedded with, that in the development of early understanding of the molecular virology of AIDS and development of AIDS vaccines, something that I worked on later after I finished my MD when I came back to Davis. So I'd come out of this lab that had been involved in retroviruses. I was very focused in retroviruses. And one of the hottest things going at the time for retroviruses was gene therapy. It was believed that retroviral gene therapy was going to be the way to go. And for me, I became totally enamored of the idea that there was going to be a gene therapist in every hospital, and this is a new specialty that I could play a role in founding.
Right.
And so I was totally wrapped up in retroviruses and vaccines and retroviral biology and gene therapy and all that stuff. And I guess I kind of overshot the target because I got an MD PhD scholarship to Northwestern. So not only did I get in, but I got a scholarship. But my wife and I were from California. We grew up in Santa Barbara area. And I can tell you that Jill did not appreciate Chicago winters, and we'll just leave it at that.
I'm Canadian, so I know exactly what she's talking about. Yeah.
Yeah, that wind when it comes off the lake is wicked. And we had a newborn, our first son, and and she just wanted to get out of Dodge. So I applied for my graduate work. I took my graduate exams and basically knocked it out of the park. I was like 99 plus percentile for my GREs, because of course I'd finished 2 years of medical school and 2 years of really hardcore biochemistry at UC Davis. So I had my pick of places, and the best place in the world to do gene therapy at the time was UC San Diego, because there are these two characters, Ted Friedman, who is the guy that originally came up with the idea in the late 70s when I was still in high school, and Inder Verma, who was eventually became head of the Salk Institute and had trained under this guy named David Baltimore. And Inder had been the one that had characterized reverse transcriptase in David Baltimore's lab, and that's what got David Baltimore the Nobel Prize. So I wanted to go to San Diego because this was the place to be. I thought a lot about myself, and I thought that I could swim in those waters, not realizing they were shark infested. So went down there, and in there, the only graduate program they had that would allow me to get access to these two guys was their biology program. So it's general biology PhD that I was enrolled in, and but really intensive. La Jolla at the time was just exploding. You had Scripps Institute. Since then, Torrey Pines Road has become one of the hubs of biotech for the world.
I used to go hiking
at that time. It was still kind of emerging.
Okay.
There was a company that had just recently been sold that had developed some of the core technology for monoclonal antibodies and hybridoma development, and that generated a whole bunch of millionaires, and something your audience can appreciate. And what I've found over time is that sometimes folks that get a flash in the pan like that, they all think that they're geniuses because they're rich.
Nothing feels like success.
Yeah.
You start thinking you're golden. Everything I touch is golden.
Yeah. And so La Jolla was full of that. And also it kind of distorted things because all of the academics saw all of this money coming in, and it kind of drove them a little bit crazy. They all wanted to get rich too.
Right.
So it was just a hyper activated, hungry environment with money and new ideas, and the Scripps Clinic and the Salk Institute, and it was an amazing pressure cooker environment that I landed in. I had I was still of the opinion that biotech was this, ivory tower on the hill, and we're all going to cure disease and do great works. Long story short, I landed in the Salk, working for Inder as the only graduate student in a postdoc lab of about 20 postdocs. Now, what that means, just to translate that for your audience, is in a big postdoc lab like that, what's done is by those that are aggressive, like Inder, is you pit the postdocs against each other. So you give multiple postdocs the same project and see who wins. It's kind of survival of the fittest environment, or you might say Lord of the Flies, is what that breeds in terms of the laboratory dynamics. And people sneaking in at night and looking at each other's lab books and all kinds of crazy stuff. But that's what goes on in these big, high-powered laboratories. At the time when I was there at the Salk, I think there was a half a dozen Nobel laureates. Renato Del Becco was there. Jonas Salk was still there. He never got a Nobel. Francis Crick was there, and it was truly the center. It was a high-powered molecular biology and virology think tank.
Okay.
With wet labs, I don't know has ever existed. Maybe the Whitehead at MIT, but and there was a lot of cross-fertilization between the Whitehead, the Salk. David Baltimore was trained at the Salk and then went to the Whitehead, and the Fred Hutchinson Cancer Research Center in Seattle. So there were these few islands of molecular virology and biology and immunology. Just to illustrate, Susumu Tonegawa, who got the Nobel Prize for figuring out B cell maturation and class switching and all that, also came out of the Salk. So a lot of really high-powered people.
Now,
and
I want to ask you,
immersed in this.
Okay.
Go ahead.
Well, I just wanted to say, you were surprised that the motivations were not necessarily about curing disease. Like, can you speak to that a little bit more?
Yeah. So this was one of the shockers for me, one of the cognitive dissonance moments that eventually, to read forward, drove me into a nervous breakdown when I got caught in patent wars and things between the university and the Salk. There's, one of the things, one of the first kind of inklings that this wasn't Kansas anymore, is that there's a pecking order among Nobel laureates. Did you get the prize yourself? How important was your prize compared to somebody else's prize? How many people were awarded at the same time? And what I found was a lot of these people are not very happy. A lot of them are kind of obsessive compulsive, not family people, not particularly well grounded. And, to get to that level intellectually, you pretty much have to be really obsessive.
Obsessed, yeah.
And then you threw, as if that wasn't enough of a fire, which I think personally is about immortality. I think that a lot of this drive is about becoming in some way immortal through literature and prizes and things like that. But it distorts people's souls.
Yes.
On top of that, you had then thrown cash into the fire. And that had just hyped things up to a level. It didn't affect everybody. It affected more of the junior faculty in the San Diego region. They were a little hungry to get rich.
Yeah.
Well, this is a fascinating graph.
That's why I'm really fascinated, because I spent some time in SoCal helping some people, and I helped some people make a few $1 million. And what really shocked me was that we had just made a ton of money. And, so there should be plenty to go around. And I was so shocked that even though we had made a ton of money for the company, I saw people get squeezed out of the company and their commissions eaten and things like this. And like you were talking about, one of the things that dawned on me is that for someone to, and this isn't necessarily everybody, but like you said, to achieve at a high level requires a certain level of obsession. And that obsession also includes all the minor marginal details. So even though there was a boatload of money to go around, easy come, easy go, so to speak. And there was I was so shocked because it just was weird for me, that cognitive dissonance, almost like what you, I'm almost hearing my own story and you talking about your experience in California, because I was so shocked by that. And I mean, we all want to make money, right? But I keep coming back to this chart because as we get news from these different agencies and that, I kind of think, to me, this speaks to not a performance-based motivation. In the jungle, not everything kills you and eats you. Some things feed on you for as long as they can. And I have a background. There's a barbell in the background here. In CrossFit, they were in legal battles for years. So they figured out how to essentially reverse type 2 diabetes. And they spent years in the courtroom fighting all these battles, and they won a bunch of these. Like this, evidence demonstrates the long history of the CDC and NIH's prioritization of industry partnerships over their charter to protect the American public. So the founder of CrossFit actually chartered an artist to make this kind of breakdown of how it works in this background, because they were just winning these lawsuits by a landslide. And I don't think that I'm not trying to say that all these institutions are corrupt to the core. That's not it at all. But I really think when humans are involved, there's a lot of women and money can do a lot to influence people. Here, this guy's winning the scientific misconduct award, the NIH, there's a lot of that. I think, so I don't want to necessarily go too deep on that if you're uncomfortable with that, but it wasn't my intent to go straight to this right away. But how do you feel that that's impacted things with this pandemic? So I'm in the Philippines right now, and I spent most of this pandemic in Vietnam. And Vietnam had no pandemic. We had our first 2 confirmed cases January 23rd. by February 6th, the whole country had shut down. All schools were closed at all levels, from public school all the way through university. We did a 2-week lockdown, and then they started doing phased rollouts. And I was living in Saigon at the time. And in March, a bar opened before they were supposed to, and a couple people end up at the hospital confirmed, and they do the contact tracing, and they found 14 cases. And one of them, early March, was on the 26th floor of my condo. My family and I ended up waking up to people with hazmat suits walking the hallway, spray sterilizing the elevator, us being asked to sign waivers saying we're going to stay in our apartment until it's safe. And the building, it was like out of movie. The building police barricaded the building. No one could leave, come in, go. And so, I mean, I was terrified at the time. And 7 days into what ended up being an 11-day lockdown for us, 7 days in, all of Vietnam, April 1st, all of Vietnam started a 3-week lockdown, countrywide lockdown. You could still go to the grocery store and stuff, but it was like events canceled, businesses closed, all that stuff. And after that, they opened up. And then almost the rest of 2020, life was normal. Some people wore masks, some people didn't wear masks. There was like surgical precision to stuff.
Yeah.
So
your story is a fascinating example. There is a serious academic study out from a think tank. I think it's the Rand Institute. I'm not sure, that I posted on Twitter recently, that evaluates the economic impact of this strategy of lockdowns, as well as the health impact.
Right.
And the data are quite clear across multiple countries. That kind of lockdown behavior does nothing but destroy economies.
Right.
And it contributes to loss of life. So that's an example of an ill-advised. So getting back to the story, you wanted the origin story. So we're aligned in that these kinds of incentives can bring out the worst in people. And I'm sure your investors are, in your community, your listeners are well aware of how money and power can drive people to do things that might, for those of us that care about things like our soul and our well-being intellectually and psychologically, it can really drive people into dark places. And so that's and not everybody. Francis Crick, to my experience, was just a noble person. And there are those that rise above this, and I want to be like them. I try really hard. And one of the outcomes of all of this is I've kind of found my own niche, and I provide services and do my thing and contribute to human health, but I really have no interest in getting on the merry-go-round. I've never sought the Nobel, although in theory I'm one of the top candidates right now. So we'll see how that plays out in the next month. So what happened was I was in this amazing intellectual stew pot at the Salk, which is basically a temple created by March of Dimes money to Vaccinology, if you think about it. And amazing building, brutalist architecture, fantastic art brutalist example. But I was a very small fish in a very large pond full of intellectual giants. And so you have to kind of, as a graduate student, you have to figure out your niche, and you want to figure out a niche that's not going to get you eaten alive by the sharks. And so I focused on the issue that was a big important one fundamentally in retroviral vectors for gene therapy, which is how is the RNA packaged? Why did it matter? Because if we could come up with ways to get the amount of virus coming off of a cell culture higher, then it would become more economically viable and also more medically viable. Then you would have higher concentration virus at lower cost. Something, again, your audience can appreciate. So that's where I was coming from, is the fundamentals of RNA packaging into retroviruses. In order to do that, I had to develop some technologies to ask the underlying scientific questions. And basically, I had to devise a way to put RNA into packaging cells that complement, that have all the necessary proteins from retroviruses to package foreign RNA and put it out as a new retrovirus. So that was the idea. And then once I could do that, then I could make point mutations in the RNA that I was putting in and ask how that affected the production of the viruses. And then I would have a good system to allow me to really ask these fundamental questions and make the overall productivity of retroviral production much higher. So that was the logic.
Okay.
And that was turned out to be a much harder lift than you would think, because no one had really figured out how to make large quantities of RNA and purify it. They were making very small amounts and then using them to make small amounts of recombinant protein using lysed red blood cells, rabbit reticulocyte lysates, and things like this. But no one had really been working on delivering RNA efficiently and making large quantities of RNA and what the structure needed to be of the RNA and what are the elements necessary to make it work. And so I had to solve all those problems. And in this pressure cooker, kind of isolated away, frantically working stupid long hours because I, had performance anxiety and wanted to live up to the expectations.
Trying to make your claim.
And my wife can attest to that, and she has. It was not a happy time for her, even though we were living in La Jolla near the water. But that's the way it was. And what came out of that was a cascade of events. I was being mentored by a postdoc in the lab that was working on gene therapy in a mouse model, and he had a problem. He discovered a problem that there's that one that happened at Vical. So this is an earlier one with Dan St. Louis that I'm referring to. Okay. And what Dan did was it's not there. I got a acknowledgement on the paper. Oh, okay. You can look it up. But so Dan found out that the expression only lasted about 3 weeks. And everybody was thinking this was some sophisticated gene regulation event. And I dove into the medical text because I'd already finished my first 2 years and finished immunology and all that good stuff. And pointed out that this was perfect for the timing for an immune response, mounting an immune response. And that set off a cascade because that was heresy. What the implications of that was that the whole idea of gene therapy was not viable, because if you're putting a foreign gene, even though it's a good gene, into somebody's body and they haven't had that protein expressed before, their immune system will reject it, just like the mice rejected the foreign gene. So for me, this was a huge paradox. The whole thing that I'd come to the Salk for and was so obsessed with, gene therapy, turned out it's not going to work, my friends. So then the question, and that's turned out to be the case. That was actually prescient, that interpretation. Now, an aside was that there was a postdoc in the lab, the senior postdoc named Dinko Valerio. This is another story your audience will appreciate. So Dinko was working on this totally new viral vector technology. It was cutting edge, 1987, 1988, 1989. It was a totally new viral vector, adenovirus. Okay. And Dinko was right at the forefront of adenovirus technology. And when he finished his postdoc, he went back to the Netherlands and started a company focused on using adenovirus for gene therapy. That company was called Crucell, and it struggled along as a gene therapy company. And then he made a decision, and he came to me at a conference, a viral vector conference, a few years later, and he said, after we left the Salk, and he said, "Robert, you're right. We should focus on using gene therapy for immune response instead of as a therapeutic. And so I'm going to change the focus of my company to be a vaccine company." Crucell exploded and was sold to a little company you've heard before called J&J. Yep. And that is actually the basis for the J&J vaccine that we all know here in the States, is that technology platform. So actually all of this tech that we know of as modern generation vaccines here in the States for COVID actually all come from Invermis lab. which
Wow.
is kind of odd. People don't think about that, but that's God's honest truth. And so in my case, I had these discoveries with mRNA delivery, and I filed patent disclosure in January 17th of 1988 on the discoveries I'd made in the fourth quarter of 87 on using mRNA as a drug. And then one of the logical applications of using mRNA as a drug was, in the gene therapy space, was vaccination. And vaccination is kind of the low-hanging fruit in terms of applications for gene transfer technology, because you don't need much protein. It doesn't have to work very good as a gene delivery system, and you can still generate a really strong immune responses because the immune system amplifies the response. So a little bit of protein is all you need. Matter of fact, it's exactly what you want.
Yeah, that's why I heard that
protein.
mRNA dissolves quickly, essentially, in the body. I don't know if dissolve is the right word, but it's also better because old vaccines are essentially built off of giving you the virus in some sort of defunct form. But with mRNA, you're not getting the virus. You're getting some
you get a fragment. It's as if you're infected, but you're only producing a small subset of the proteins. Now that turns out to be a problem in the current situation.
Yes.
Because they're only expressing one of the proteins.
Okay.
So you mentioned old school vaccines. Actually, the oldest school vaccine is the smallpox vaccine based on cowpox. Okay.
Okay.
And it actually goes back, it goes back to Jenner, but it goes back to China, when they would take smallpox lesions and little scabs, and they would blow it in the noses of patients to inoculate them. So that's actually mucosal vaccination, by the way, what they were doing back in the day. So the thing is that with our modern smallpox and polio vaccines, the vaccines that wiped out polio, those are live attenuated viruses. Same with yellow fever. Okay, yellow fever is a live attenuated virus.
I got that.
And those vaccines work really good. They're very durable. They're good for eradicating disease. What do they provide? A broad-based, robust immune response that covers all of the proteins manufactured by the virus for the most part. Okay, so to read forward to our present situation, where just a couple of weeks ago, it was revealed that natural immunity is about 20 times more effective than vaccine for providing protection against SARS-CoV-2. Well, that kind of is like the old school live attenuated vaccines that work like gangbusters. But the problem, that's an old term, sorry, before your time.
No, gangbusters is good. We're good.
Okay.
So let me just recap there. So you're saying that natural immunity is 25% better, stronger, longer lasting overall versus these COVID vaccines that we're getting now.
That's truth. And as measured by protection from disease and infection. So they're about 20-fold better, and they're broader, and they have more durability. That's a fancy vaccine word for how long does it work.
Right.
And one of the things that's become clear with the Pfizer vaccine is the durability. I've been fact-checked on this and told that this is a false statement.
Efficacy. It's diminishing drastically.
Okay, yeah. So we call it effectiveness. Efficacy is in a clinical trial. Effectiveness is in the field. Just the technical terminology here, we got to get this right, or the ankle biters will be at us.
Sure.
So those data are clear, and that's one of the things. There's a few events that have come out that have really scrambled the narrative that has been pushed by public health across the world, but particularly here in the United States.
Very much.
One thing that your listeners will appreciate, the United States has been seen as the world leader intellectually. In this space. And so there's been a whole lot of deference, let's say, there's a nice word, deference to whatever is coming out of Tony Fauci and the CDC. And time has proven that has not, those statements are often wrong. I like to say Tony Fauci is like a surgeon, often wrong, but never in doubt. So maybe he should have been a surgeon. They've been willing to put out information, starting with the mask flip-flops all the way through, that's based on personal opinion rather than based on data. Here's another, while we're at it, little thing that I'd love to get to your investor community. I've lived and been at the tip of the spear through way too many outbreaks and pandemics and epidemics. H1N1, Zika, Ebola. I was the one that brought the Merck vaccine to Merck. Not this one. This is before my time.
But this is the reason why I heard that this is the outbreak why vaccine companies can't be held liable, because after this and after all these people came, this is the one that caused them to now say, hey, if we're ever going to do vaccines in the future, we don't want to be held liable. And the lobbying and the elbow rubbing around this is the one where this
is what set it up. And I'm sure your investors will appreciate if you create a situation where the risks are assumed by the public and the government, then the incentives for a given company to be mindful of the risks become greatly diminished. And you can make a ton of profit in an outbreak. They are extremely profitable. I argue it's war profiteering. That's right. But that's the truth of it.
Right.
And we've seen how many billionaires have been created during this outbreak.
Right. Well, and that's some stuff I want to talk about, because this is where, like, vaccines work, right? To an extent, the vaccines work. There might be complications with it, but or you think they don't work at all, and that's okay. I'm just trying to get that question put out there.
Yeah. So my position is on this, because it's a little nuanced. The press wants to push us into you're either pro-vaccine or you're anti-vaccine.
Right.
And there's a lovely article out in Forbes that talks about the noble lie and the loss of the middle. And I reside in the middle space. My current hangout, if you appreciate the terminology of modified limited hangouts, my current hangout is that the data clearly demonstrate protection from death and disease that provides clinically significant benefit in aggregate to the elderly, immunocompromised, morbidly obese, and those with other risk factors. That it's important to understand, we get lost in the fear porn that is promoted here. But it sells newspapers, it's clickbait, and it's what drives the legacy media, and a lot of the modern media is headlines and articles that generate fear and other strong emotions. And that is not very conducive to accurate dissemination of information.
No, right now there's a huge, forgive me for interrupting. I know you've got so much knowledge on this stuff, and I feel like I want to get some of this stuff to the forefront, where exactly what you're saying. Sorry, I interrupted. Now I'm taking a pause, but I think like you're saying the vaccines are good in certain use cases, but not all use cases. And right now there's a lot of misinformation
because the risk of severe death of severe disease and death for almost everyone, especially children, but almost everyone up to the age of 60 and even beyond. The risk of death from this disease and it is in the range of 0.00 2 or 1%. Okay, so if you get infected, on average,
right,
all of the death and disease is clustered in the elderly. By the way, that's the same truth with influenza. Influenza takes out the really old,
right,
just like this disease does.
right.
This disease is more efficient at taking out the old. Now, I'm going to just illustrate this kind of give a gift to your audience by sharing some of my own medical history. Okay, so I don't really like to do this very much, but it illustrates the point. I was infected in the Biogen outbreak with COVID, with SARS-CoV-2, and I developed COVID. So what that means is I was at a conference in Boston and physically staying across from Biogen headquarters. There's an old remodeled fire station there that I was staying in, and walking to MIT for this conference on drug discovery and AI and ML. And I got infected, came back sick as a dog, discovered famotidine by self-treating as a therapeutic, and had a pretty rough clinical course. Couldn't walk up the hill without having to stop for breath. And so I don't want to minimize the nature of this disease if you get it.
Right.
And the disease is your body's hyperinflammatory response to the virus. It's
right.
not actually the virus is the primary cause. It's not virus damage. It's your body's reaction to it that drives a lot of this disease.
Okay.
So in any case, in my case, I developed COVID and I developed long COVID.
Right.
I've had it ever since. I recently completed a course of ivermectin that's helped me quite a bit, in my opinion. And despite all that, no, I didn't take the horse drug. My horses take the horse drug every other month, but I did not. I took the human form. And I recently had a full pulmonology workup with pulmonary function tests, which I expected was going to be horrible. I'm totally within normal limits, got a clear, clean bill of health on my pulmonary function, and I'm good to go, and I may actually live for another 20 years. So yay.
Good. That's good news.
Point is, my wife was really relieved. So the point is, yeah, this is a bad disease. It's not the flu, it's worse, but it's not going to necessarily put you in the ground, especially if you get early treatment.
Right.
Okay. So now another thing that's contributing, there's all kinds of variables that are contributing to the mortality rates in the States, which, by the way, are among the worst in the world.
Right. They are the worst in the world.
They're way over treating with dexamethasone.
Yep.
They're blocking, there's been active blocking of early drug treatments by the NIH and the FDA and the CDC.
Yes.
And a lot of us are just going nuts trying to figure out what are they thinking. So Merck, ivermectin is one of the safest drugs in the world.
Well, that's what I want to mention
medicine.
for a second. This study shows, what was it? They were getting something like 60 milligrams. I saw it. Maybe I don't want to dig through it while we're on this call, but this is a study. I think they were getting 60
so 150.
milligrams.
You just showed it. Okay, you just showed the dose. Okay, Scroll down.
150.
Scroll down. 150 to 400. Okay, 400 is a wicked high dose.
Yeah.
The standard dose for humans and for horses, by the way, is 200 mics per kg. Okay.
Yeah.
So, 1 mg per kg is a wicked high dose, and it's safe. So all of this jibber jabber about which Merck put out. I mean, the thing talking about disclosure of conflicts of interest, Merck has licensed a drug developed at Emory under Defense Threat Reduction Agency sponsorship that is a direct-acting antiviral and they paid a bunch of money to Ridgeback for it. Ridgeback had licensed it from Emory. DOD had paid to develop it. Merck licensed it, then wouldn't play ball with us at the DOD. We wanted to try testing it. They put out this press that ivermectin is not safe.
It's not safe,
it's clearly false.
It's an animal disease. That's why I want to show this. For those that can't see the visual, we're on Merck's own website about a 30-year donation program where they were donating mectizan, which was ivermectin, and they distributed more than 300 million. They distributed free doses to more than 300 million people with more than 4 billion treatments donated annually.
Yeah.
And so that's where we're in this propaganda thing. And I shared another thing here where we were talking about emergency use. The FDA may authorize unapproved medical products or unapproved uses of approved medical products to be used in an emergency to diagnose, treat, or prevent serious or life-threatening diseases or conditions caused by CRBN threat agents when certain criteria are met, including there is no adequate, approved, and available alternatives. There's no one making money on ivermectin right now. It's out of patent. It's been around for 75 years. There's no money in it, and they can't have these emergency use of these vaccines if
so
there's something valid.
The same is actually true of hydroxychloroquine. And there's people are using hydroxychloroquine all over the world. I'm about to join I'll travel to Rome next week for a meeting of physicians from all over the world that will be developing consensus protocols for early treatments. And most of those are based on either ivermectin or hydroxychloroquine.
Yep.
There's a legal case proceeding that I can't talk about that relates to what appears that there is evidence of conspiracy within the government to shut down both ivermectin and hydroxychloroquine as early therapeutics. The government actively blocked the development or allowance of any early therapeutics. So I was just speaking to some lawyers yesterday that I'd been supporting on another case involving regulatory capture and vaccines. And what was observed in that case is child's play compared to what I'm seeing happening here. The regulatory capture that has occurred, and it's really governmental capture, as you know, embodied and enabled in contract terms that, for instance, have been leaked from Pfizer, is profound. And the information control and horizontal integration that has enabled this information control is never been seen before at this level. And so we're, you know, the ivermectin push stating that this is a horse drug and you've got to be crazy to use it.
No, this is a study, ivermectin, a systemic review of antigen. Is this the one I think it reviewed? Is this the one they reviewed 50 years?
There's 4 or 5 of them out there.
Yes, this one. So it's like, it's proven effective. Several studies have reported antiviral effect of ivermectin on RNA viruses such as Zika, dengue, yellow fever, West Nile, Hendra, Newcastle, Venezuelan equine encephalitis, chikungunya, avian influenza, chikungunya, respiratory syndrome, human immunodeficiency virus type 1, and severe acute respiratory syndrome, coronavirus 2. Furthermore, there are some studies showing antiviral effects of ivermectin against DNA viruses, such as equine herpes type 1, BK polymavirus, pseudorabies, porcine
circovirus, and bovine herpes virus. So there's the problem, the logic, and we tried to get at the DoD. We tried to get allowance. What we now have allowance for is to proceed with testing the combination of famotidine and celecoxib in both outpatient and inpatient studies. So that IND finally cleared, but we had wanted to also test in rigorous double-blind randomized clinical trials the combination of famotidine, celecoxib, and ivermectin. And we have outpatient data and laboratory data supporting it that that combination is even more effective. But the FDA insisted that we would have to demonstrate the mechanism of action of ivermectin in cell culture before they would allow this trial to proceed, which would have cost us another, 6 to 12 months.
Well, it's like legal battles. The deepest pockets can win because they just stretch it out. They reschedule it. They make you show up to the next one. So let me ask about this. So there's clear censorship going on. You said there's lawsuits in the works. We just saw, if those that didn't see, there's been billion-dollar settlements for these companies that have clearly done criminal and civil offenses, from kickbacks to marketing drugs that are known to be dangerous. What should have, I guess I should ask, what, as the inventor of the technology, what would have needed to happen? What would need to happen for it to have been fully ready for humans to take it? The mRNA vaccines that are the COVID vaccines that are out right now.
And let me just restructure that just a little bit.
Sure.
Here in the States, we have 3 vaccines that are either emergency use licensed or tentatively market authorized. The BioNTech product is not actually on the market yet because it's the same as the Pfizer product mostly, but it's technically a different product because it has new labeling and packaging and things like that.
Okay.
So really we have 3 vaccines. They're all genetic vaccines. They're all based on gene therapy technology. They all express exactly the same protein for the most part, which is the spike. And it's the intact spike with 2 amino acid modifications in the receptor binding domain that lock the receptor binding domain. Fancy words for your audience. That's the part of the spike protein. The purpose of the spike protein is to allow the virus to bind its target cell and to insert or kind of inject the genome of the virus into the cell that's being infected.
Right.
So that's what spike does. And so it's got these 2 functions, and it undergoes a structural change in order to do that. I like to use my 2 hands. There's actually 3 subunits that come together. So if you think of my arms as the stalk, and then there's this head bulbous area. You see it on all the pictures of a coronavirus.
Right.
And represents spike. And the binding part is out here on my fingertips that recognizes the receptor that it binds to. And they move around, flop around, open up, go upside down. They move all around.
Yeah.
And what's been done is to insert a 2 amino acid mutation into spike that makes it so that instead of flopping around and undergoing this conformational change for fusion with the virus, it locks it into an open conformation. And it was based on the assumption that the antibodies that will inactivate the virus bind, if you can think of this as catcher's gloves.
Right.
If that works for most of your audience.
Right.
So there's a pocket in the catcher's glove, and that's the pocket that the receptor, that the protein on the cell binds to. So think of it that way. That it's like a fielder's glove. It's more like a fielder's glove because it can be open or closed, right?
Right.
Okay, and so think about the guy in the outfield with the glove that's got big fingers and it's open or closed,
right?
And so that when the ball hits it, you close it up and you don't lose the ball, right? So it's kind of like that. Only they've introduced a mutation that's locked it into the open conformation, so the glove can't close.
Okay.
And the assumption was that the antibodies were going to have to hit the binding pocket. So that's the pocket in the glove, right? That would stop. And so if they hit that binding pocket, then they'll make it so that the virus can't bind onto a cell. That's the whole underlying logic of all of these vaccines.
Okay.
Is they are exclusively trying to target inactivating antibodies from B cells that'll bind to that pocket in the glove and make it so it can't catch the ball, basically. Right. Okay, if you think of the ball as the receptor on the cell.
Yep.
So, okay, so this isn't going to work for your European audience that plays cricket, but hopefully people will get it.
That's okay.
So it turns out that's not true. There's been a ton of work since looking at monoclonal antibodies and various antibody preparations and where they bind on spike. And for the most part, they're binding here, right at the tip, or they're binding the backside, or they're binding at the junction between two spike proteins, okay?
Right.
So the whole idea for the mutation that was introduced, people say, oh, that mutation was introduced to keep spike from being toxic. No, in order to have done that, the people doing the bioengineering would have had to have a time machine, because it wasn't known at the time they were doing the bioengineering about these other toxicities associated with spike that we now know about.
Right.
Okay? So, and the FDA never forced Pfizer at all to go back and prove that the engineered spike was safe and did not have the biologic activities that the wild type spike has, the wild type being the kind that's actually on the virus. So just to disambiguate.
So
we don't actually know whether and I've been fact checked on this, but they kind of don't get the point. My statements in the Brett Weinstein podcast was that spike is a toxin. That is true.
Right.
It has been proven again and again and again. Native spike is a toxin. And so then they say, well, but the geniuses at NIH engineered it so it's not going to be a toxin. Well, that's interesting because they've never shown that. They've never said that was the reason they were doing it. And they would have had to know and acknowledge that it was a toxin way back then in order to be able to foreshadow that they would have to make this engineering. Last point on this thread is that with pharmacology and pharmaceutical development, I like to say that the rules of the French judicial system apply. You're guilty until proven innocent. And so you can't assert that that a given protein is safe and effective unless you do the hard work to show that, in fact, it's safe and effective. So I don't know why you're pulling these. These are other examples of issues.
And that's why because I want to ask, are these at all real concerns? So I feel like what you're saying is the vaccines help to fight this version of the coronavirus, but it might, what's the term? Would it make you, is it hypersensitivity? What's the term? Vaccine-induced disease enhancement? Is that a real thing? Is that something?
Thank you for using that. Thank you for using that term instead of antibody-dependent enhancement. Antibody-dependent enhancement is a subset of vaccine-enhanced disease and replication, which is the vaccinologist's worst nightmare. It's what we ran into with all prior attempts to develop vaccines against these coronaviruses, with two exceptions, those both being veterinary vaccines, both of which are administered intranasally. Yeah, and you can actually find clips from Tony Fauci warning about ADE, and the ADE warnings are written into the federal, the FDA comments that were the basis of the emergency use authorization. There have been no rigorous studies done about whether or not vaccine-enhanced disease is occurring. Despite the FDA saying that they should be done, the vaccine developers have chosen not to do them. The FDA hasn't forced them to do them. And though there is the one thing that your listeners need to understand is that it has become wicked hard to publish anything that goes against the dominant narrative.
Right, the dominant narrative.
So that's another thing that's come out, is that almost all of these publications that we've relied on to be providing objective truth are dependent on pharma money for advertising and sponsorship.
And pharma, we've
so,
seen has just almost annual lawsuits of hundreds of millions of dollars. So you just to say that they've never been able to.
So
sorry.
You can't publish this stuff. It's just wicked hard, and so that's why you're not seeing it come out in the literature. And if you're an academic, why would you spend 6 months of heavy lifting and scarce dollars on trying to demonstrate something that you're not going to be able to get published? It's crazy. You wouldn't do it. It's just, your investors understand this.
I want to clarify something you said before. You were never able to create a safe vaccine for a coronavirus in the past, except for 2 exceptions.
Feline infectious peritonitis and bovine coronavirus are both veterinary vaccines. And they're both considered kind of crummy vaccines by the veterinarians. But those 2 did manage after a long, one of the things that's nice about veterinary vaccines that you can't do with humans really, is that you can challenge the animals with lethal doses of virus, and it's ethically acceptable. It's obviously not ethically acceptable to challenge human beings with lethal doses of virus. And the truth is that because of the nuances of the ACE2 receptor and the TMPRSS protein that activates the virus once it's bound, because of those nuances, humans have different affinities in their ACE2 for this virus than other animals do. And so basically, we don't have an animal model that's really worth much. The phrase "a tinker's dam" comes to mind, other phrases too. So we can't do the challenge studies in the way that we would love to be able to do, because we end up, just like with AIDS, with very artificial systems, like using the virus that Murray and Preston Marks identified years ago and related viruses to challenge monkeys, or human, basically gain of function versions. To do those studies, and they're still not conclusive, and we still don't have an AIDS vaccine. So in this case, I don't want to give your investors a sense that there's no hope on the horizon for a decent vaccine.
No.
These vaccines are first generation, and they, in their rush to get something out the door, they focused on the low-hanging fruit, which was spike, as a single antigen. And there are multiple other vaccines in development, and some even licensed. And full disclosure, I'm involved in one of those being advanced in India by a company called Reliance, owned by a gentleman named Mukesh Ambani. You can look him up. You'll find he's one of the wealthiest people in the world, and Reliance is the largest conglomerate in India. Something that most Americans don't recognize is that there's actually a huge financial horizon in India, and their Indian vaccine developers are really, really good. I think that the vaccines industry and pharmaceuticals industry in India will come to dominate global supply over the next decade or so. So that's a news you can use. But the team that I've been helping coach at Reliance now has obtained allowance to begin phase 1 studies. And that vaccine, just to give an example, is a much more traditional vaccine. It's manufactured in mammalian cell culture and in E. coli in bacterial culture. It's purpose designed to be extremely low cost, because Mr. Ambani wants to be able to vaccinate the rural poor in India. And, 1 billion people at even $10 a dose, that adds up to some real money.
Very, very fast.
Right. So that's his goal, is to be able to provide a product that will be safe and effective. And in that case, that product expresses 2 different proteins. It's a subunit of spike, the receptor binding domain. It's not the whole protein, so it doesn't have these biologic activities.
Right.
And it's also a subunit of one of the other core matrix proteins, the nucleocapsid protein associated with the virus. And so it has a broader antigen range, and we'll see how it works.
Yep.
And it's adjuvanted with more traditional adjuvants, alum and CpG. So that's something, that's one of many examples of second generation vaccines that are going to be coming onto the market. And in terms of, I don't want to pump, my full disclosure, I'm on retainer to Reliance Vaccines.
Sure.
So I'm not pumping Reliance. They're one of many examples, but if your investor community is looking for new horizons, they may be wanting to look at next generation products, not necessarily at these products. I think there was, as I recall, there was an announcement by Pfizer to stockholders that they should expect that this current vaccine loses its effectiveness at something like 6 months from now.
So vaccine only lasts for about 6 months. We're almost at the top of our time.
Well, I'm talking about not only the durability for you, I'm talking about the overall efficacy of that vaccine will fall below acceptable limits because of the escape mutants that are being generated.
Right. There's over 100 variants right now, right? There's something like 5+ variants a week they're talking about.
So we now know that this virus is mutating at a higher rate than we had expected.
Right.
And there's some nuances here. The thing about vaccinating into an outbreak when you already have a whole lot of people with virus is that what you do is you got to kind of remember Darwin and natural selection.
Yeah, evolution.
You'll select for escape mutants.
Right.
The easy way for everybody to understand this is the overuse of antibiotics in the livestock industry.
It's created superbugs.
Is correct. Okay, so you can think of this as we're creating a super virus bug through the selective pressure of mass vaccination when we don't need to vaccinate everybody.
Everyone. Right.
We really only need to vaccinate the people at highest risk. And then the virus would be subjected to the natural immune systems of everybody else, where you have a probability of dying of, a tiny fraction of a percent.
Right.
And that is only that high because the government, in its infinite wisdom, has seen fit to make it impossible for docs to actually practice medicine.
And actually try different treatments.
Yeah, recognize and start implementing them. We've been trying them for 1.5 years now. We know a lot of things that work. The situation, like with famotidine and celecoxib, famotidine is over the counter. You can all buy it. It's called Pepcid. And celecoxib is called Celebrex. A lot of you use that for your arthritis. So the combination, and it turns out that Celebrex blocks COX-2. By the way, aspirin blocks COX-1 and COX-2. We don't recommend aspirin because there's been some history of adverse events in respiratory diseases with aspirin, and it's basically blocking too much of your immune response.
Right.
But there are papers out there showing that high dose famotidine, so high dose Pepcid plus aspirin, actually is pretty effective.
Right.
So there's a bunch of these drugs. The government has actively blocked their deployment and blocked the ability of physicians to use them. And it seems that the logic is that they perceive them as competitive with vaccination, that if these drugs are available, people won't accept vaccine.
Right.
Well, because why would that be? Well, because the vaccines aren't perfectly safe and they aren't perfectly effective.
Right.
One of the things that's kind of become increasingly apparent. So that's kind of where this is going, and it's unfortunate that we've all been blocked, but we probably have. There are credible estimates of hundreds of thousands of Americans unnecessarily dying because of mismanagement of the clinical disease, point being that rate of death in the average population
right.
of something in the range of 0.002 0.001 percent risk of death if you're not in the super high risk groups, that's actually artificially high because we haven't been able to deploy early treatments. If we had, it would be lower, and that's part of the reason why here in the States, our death rate, our mortality per case of hospitalized COVID is higher than just about anywhere else in the world.
Right.
What
So
we should be doing is keeping people out of the hospital in the first place. So any of you that have had this experience or your relatives, what's been happening is you go to the emergency room, you say, doctor, I'm sick. I can't breathe very well. And they test your blood oxygen and they say, you aren't yet at the threshold of having blue lips. Go home and come back. And you say, but doctor, is there anything, come back when your lips are blue? Functionally, that's, having a blood oxygen lower than 80.
And that's it.
Or
Go to the
lower than
hospital when
90.
you're on your deathbed.
Yeah, exactly. And if you haven't been vaccinated, I mean, there's things like the Toronto Star headline the other day that basically say, well, if they weren't vaccinated and they come to the hospital, you should just let them die.
Right.
I'm shocked by all of this, but the truth underlying all of this is that there have been good repurposed drug therapies, and American physicians are blocked from using them. They're blocked from being able to practice medicine under threat of losing their license. You're only allowed to practice in the way that the NIH says you're allowed to practice.
Right.
So that's where it's coming from. And essentially that equates to what Tony says is what goes.
Right.
Tony doesn't practice medicine. Tony's never treated a COVID patient. We got to get that through our brains. Okay. Well, and Tony is subjected to the incredible conflict of interest relationships with key stakeholders all across this space, particularly in the vaccines industry. He's tight as a tick with Bill Gates. And we've got a whole, Facebook and Zuckerberg is invested in vaccines.
Yep.
Bill and Melinda Gates Foundation their core portfolio is invested vaccines. The Robert Wood Johnson Foundation is one of the largest shareholders of J&J. These entities that we think of as benign and not having conflict of interest that are funding a lot of this propaganda campaign and these fact checkers and everything else, they all have significant conflicts of interest.
For sure. Well, again, we talked about the settlements of these pharmaceutical companies, and if they're paying $2 billion, $3 billion, how much money does it cost to buy someone? I mean, how much money would I have to give you to get you to say your tap water made you feel ill? You know what I mean? Or like some sort of shampoo made your testicles smaller.
My wife always says to me, if Pfizer had only realized how cheap you could have been bought, then they would have had none of this headache.
Right. So this is something, again, people are concerned. Should I get vaccinated? Why? That's really what a lot of people are concerned about. We haven't been able to get vaccinated because Vietnam didn't think they needed them and they were slow to get them. And now in the Philippines, you have to book like you're on schedule and all that stuff. But they're pushing everyone to get vaccinated.
Hold it. You're a great case study. Let me dive into that because I had a long conference call with a colonel that represented a group of 20 nations or entities in Latin America that have ties to the UN. Okay. And it was quite a tense phone call. He was pressing me. It was basically a consultation to help them devise policy. And there was translators involved, and I had to be on my tippy toes intellectually. Because I knew I was going to be influencing Latin American policy. And he made the case that it's not in the interests of these Latin American countries to vaccinate. The vaccines only last for a few months.
Right.
They require cold chains that they don't have.
Right.
Pfizer in particular has quite egregious contract terms.
Right.
And they just don't see it as making sense in emerging markets. The counter thread is that the Western nations are basically hoarding all the vaccine and super saturating all of their populations with vaccine unnecessarily, and not making it available for the high-risk groups in the rest of the world. So there's a lot of tension in this, and I got to say that the positions that Vietnam took and other emerging economies are taking, emerging economy nations, that they really don't want to buy into this carousel with the vaccines. There's a merit to those arguments, and I have to acknowledge that for those situations, there's a logic there that's sound. And now that we know that natural infection provides superior immunity, the idea that many of them are focusing on, many of these, for instance, Mako, ivermectin is over the counter.
Yeah.
And it's cheap.
Got it in the Philippines, got it in Vietnam, got it in the Philippines. Yeah.
Yeah. So there are good protocols out there that can be used for early treatment. And if you get on this thing early, you end up not going to the hospital in the first place.
Right.
So that kind of makes sense to me. And why we're not doing it here in the States, I just don't get it. And a lot of people are perplexed about this. And that leads to all the conspiracy thinking and the regulatory capture observations, because there's just so many things that don't make medical sense. They only make financial sense for the vaccine companies. So that leads to people speculating about, their minds go off into Klaus Schwab and the World Economic Forum and the Great Reset, and we can go down that rabbit hole for another 2 hours if you want.
Sure.
Not that we have time today, but there's a whole lot of stuff there that active minds trying to make sense out of the nonsensical legitimately will go down, trying to investigate those various alternative explanations. I try not to do that. I try to stay, based
Focus
on the facts that I can demonstrate.
Yep.
Yeah. It's not my job to. I'm venturing into public policy a little bit with the op-eds that Peter Navarro and I are putting out in the Washington Times, but for the most part, I try to stay away from those. But for sure, in terms of emerging economies, there's a logic to saying, not for me, thank you very much. Now, here's the observation, and this is another one of the conspiracy Legos. There's a group of, I think, 5 different national leaders in Africa that have all said, made clear statements that they don't want to take vaccine, every one of which has been assassinated. Now, is that just circumstance or not? I don't know. I don't know African politics. I don't know what's going on there. But there's certainly, for those that are conspiracy minded, it's an odd coincidence.
Well, we don't even have to label them conspiracies, because there's just like real facts, and certain things are just very fascinating and interesting. And that's, having been involved in having created the baseline technology, that's why I'm really curious to know about this. Because if I did jujitsu for 6 years, trained with Hickson Gracie and a bunch of others, and and it's all about kind of, you do things to know their next move, right? So it's like it's almost like Wayne Gretzky said, when I play hockey, I go where the puck is going. And so there's all this push to cause everyone to get vaccinated, and these people disappearing and all these things like that. And it's not to try to get so much into it. It's to talk about the real facts. That's why I tried to use Zoom to show some stuff about, that the States is leading the way on this.
Yeah.
But they're clearly in it for profit. If you look at our life expectancy versus, I know we're up for time.
But let me leave you with one thing that relates to this thread that your audience will appreciate, I think. One of the things I've observed again and again and again through these outbreaks is that the WHO and CDC and public health leaders generally operate only on what they consider to be validated data. Now, that's starting to break down, in, like with the triple jab, they're starting to get way out on the edge on things that they really don't have data for, and I think are risky. which is part of why there were these resignations at the FDA because of that.
Yeah.
But in general, what you see, if I can put it in terms of your investor community, it would be as if someone only makes investment decisions based on what they consider to be fully proven data. Now, your investor community will appreciate that if they were to do that, they would get their clock cleaned every single time by momentum traders and those traders that are using a totally different approach, which is basically risk mitigation, looking for leading indicators, and watching for black swans. This is language that your community can appreciate. I know you can. Okay, I advocate that if there's anything we learn from this, it's that the World Health Community needs to start acting more like stockbrokers. They need to think like stockbrokers. They need to be looking for leading indicators. They need to be performing risk mitigation, and they need to be constantly surveilling the landscape for black swans, not 6 months behind the data because they're only going to react to things that are validated.
Right.
And that, I think,
at that point, yeah, you make money by being right, and by being right, you can't wait for success for census. I forget, Naval Ravikant had some quote like that, like you win and make money by being right first, and to be right first, you can't wait for everyone to agree. Yeah,
yes, precisely, and that is how we need to manage global public health in the time of outbreak crisis. Precisely, we need to think like stockbrokers, and we need to put the infrastructure in place like the investment community has. I mean, think about the massive investment that's been made in AI, ML, and all the other toolkits to allow your investor community to make accurate predictions. Okay, why don't we have? We ought to have 10 times that infrastructure for public health.
Right, and that's where I don't. How do we not know? How do we not know they already do? And that's partially why things are being pushed. And I don't want to go down the conspiracy rabbit hole. I do want to ask.
I can tell you as an insider that they don't.
Okay.
There are movements afoot, and there are some. The CIA has advanced programs like Argus to try to do this. That's data mining, but it is just like with the investment community. The term is data fusion. The data come in from a whole lot of different kind of data streams of different formats, and it is wicked hard to integrate all of those into a comprehensive package, which is why folks like your community hire people like me, is to try to make sense out of all this stuff, because the only way you can really do it still, the ML DL stuff isn't good enough yet to be able to integrate all of the different disparate things that a human being, a human consciousness that's experienced can process. And they're not, the computational tools are not yet sophisticated enough to incorporate a lot of these risk scenarios.
Right.
But they may eventually get there. And, God bless us if they do. But by that time, the robots will rule the world, I think.
Right. Well, Elon Musk announced the Tesla bot. I don't know if you heard that. The Tesla bot, the iRobot bot's going to be here soon enough. He said it's not going to go faster than 5 miles an hour or something, so you could outrun it if you had to. Yeah.
No, if you hang out, it's fascinating to parachute into Silicon Valley. It's just like in DC. In DC, the ads are totally different. They're all about selling big contractors and beltway bandits. And if you parachute into Silicon Valley and listen to the radio, you get bombarded by ads for very sophisticated issues relating to the latest trends in computational technology and deep learning. And people in these different sectors have insights that are not generally available to the rest of us. But I guess that's why you do your podcast.
Right.
Now we're 15 minutes over, pretty close.
Yeah. I want to try to ask 1 or 2 rapid fire questions before we get off. And so one is, you aside from the vaccine escaping, or sorry, aside from the virus escaping the vaccines, could the mRNA spike protein like cause issues with like prion diseases? Can it pass? Can the lipid nanoparticles, can they pass the blood-brain barrier?
So those are the honest truth is those are risks. We talked about risk mitigation and risk identification. And normally what's done is that we take the time to run those risks to ground before we start jabbing people.
Right.
But in the heat of battle, under the pressure of overloaded hospitals, we decided that it was okay to roll this out without solving all of those questions. And if you take as a working hypothesis that the vaccine encoded spike behaves akin to the natural spike, then you have to conclude that the next logical step in that is that natural spike does open blood-brain barrier, will let things get across into the sanctum sanctorum that normally are prohibited from going in. And the spike is cut loose from the expressing cells, and the vaccine developers were not forced to characterize where the product, the active drug product is going, and what cells it's delivering its payload into, and how long they make protein, and how much protein they make. None of those are known for any three of all three of these genetic vaccines.
So it sounds like if you don't need to take it, don't take it.
That's my logic, is that there are adverse events that are well documented, and it's important to remember that those adverse events that are documented have to pass through a filter. They have to be clinically significant. So, for instance, if you're a high-performing athlete, you may notice that your peers have a drop in their performance after receiving vaccine. And if you're out on the edge of your own performance, say you're a 30-something NFL football player, you may say, hey, I'm close to retirement, and the last thing I need to do is take a jab that's going to drop my performance by 3% or 5%. Those kinds of performance changes aren't clinically significant. So they don't show up in the CDC database, but people observe them. So that's why you've got footballers and track stars and such like saying, no, I'm not going to take the jab, because they are seeing in their peers modest decreases in functional performance after taking the jab, things that are subclinical. The other thing about a lot of these adverse events, it's right in the FDA's authorization letter for the BioNTech product Comirnaty, is that the CDC now acknowledges that their databases are not sufficient for detecting rare adverse events. They say it flat out. So this is something that I had said on Tucker Carlson a couple months ago, and everybody got their tails in a twist because Tucker just wants to take down the government, and Robert was supporting Tucker, and blah, 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 blah. Well, now it's actually written into the authorization letter that the very thing I disclosed on Tucker, which is that the VAERS database and the other CDC databases are inadequate for assessing these adverse events. It's now officially in the FDA licensure package. So within 2 months we went from crazy Robert is throwing bombs that are fact checked to a federal document that validates everything I said, and that's happened again and again. So the problem is that with your question about should I take vaccine, the honest truth is I cannot point you to robust documentation that demonstrates what the spectrum of risks are. I know, and it's in the literature, that the risks are much broader than the ones that the FDA officially recognizes.
Right.
For instance, one of the more common ones is viral reactivation. How does viral reactivation happen? I don't know. It seems to be some inflammatory messaging that's happening post-vaccination. Can I assure you that the lipid nanoparticles aren't affecting ovarian performance or aren't affecting placental performance. No, I can't, because we don't have the data. And anybody at the CDC who claims otherwise is lying. I mean,
Right.
they're misleading. We can use whatever euphemism you want.
Right.
But they don't have the data to support that, so they shouldn't be saying those things. And so I can't say to you, hey, here's your risks for vaccine based on, if you fill out this little thing on your personal device, and then I can run it through a machine learning tool and give you a risk of what are your risks for adverse events and what type. I can't do that because the data don't exist to support that app on your cell phone. Now, there are apps for your cell phone that I've seen that are based on deep learning and machine learning data that can very accurately predict your risk of severe disease and COVID. Okay, and that's part of the toolkit that I'm recommending be deployed, so that you can accurately judge for yourself at least what's the true risk for you personally to developing severe disease, being hospitalized, etc., if you were to get infected by the virus. Right. That's the kind of stuff that's available. Why we haven't had it distributed, that's another one of these big questions of, what are they thinking? Well, so I hope I've run that question, should I take the vaccine, to ground. The answer is I can't answer it. Right. I can answer it really clearly. Go ahead.
If there's a synthesized misfolded protein, how would we know? If we were synthesizing,
we wouldn't, because the FDA has not forced the developers to do the heavy lifting in the rush to jam this out. And with all of the public messaging and PR, keep in mind that all of that propaganda that's being blasted out to the general public is also being received by all the bureaucrats and the folks at CDC and FDA. And they know darn well that their supervisors don't want bad news. So, if you don't want to, it's just shown by Marian Gruber for signing, because she got into a cat fight, apparently, with the CDC over the third jab. The way this works is you get in line with the diktat and lose your soul and your professional integrity, or you resign. Those are your options. Wow. And if you're working for the federal government, your primary driver is security. Right. Okay. You wouldn't take a job at 80% a market rate if you weren't focused on those bennies. Right. So all of this, I think a lot of this, we don't have to go to Klaus Schwab and the Great Reset. I think a lot of this we can explain by financial incentives, regulatory capture, and structural disincentives that exist within the system that ought to be fixed.
So is there another way to end this pandemic in a faster method than trying to rely on these vaccines?
Well, you can look up in the Washington Times. Peter Navarro and I have put out 2 op-eds to this point. And then I just did a long interview with Epoch Times. I know it's Epoch Times, and some people discount that. It's now the 5th largest publication globally. So that's coming out in American Thought Leaders. There's the first part of that interview just dropped last night, and you can find it. It is extensively footnoted. The references are in there, and what Peter and I have laid out is a 4-pronged strategy of vaccinate those that are at extremely high risk, provide apps that allow people to assess their risk so they can make their own decision about whether or not they want to take the jab. So empower people to assess their own risk. Make available early treatments. And the other part of this, in order for early treatments to work, we have to have home diagnostics. And they have those in Australia, by the way. The FDA and Tony Fauci have blocked their development here in the States and licensure, but the Aussies have them. and it's a quick diagnostic based on antibodies and other things, and you can tell whether or not you have been infected. And what we need is a rapid diagnostic home test that with all of the leakiness and flaws and false positives and false negatives that'll come with that strategy, but at least make it so that people have got to be able to be identified early on in the course of their disease, that they have the disease, so that you can begin therapy early on and keep you out of the hospital in the first place. So 4 things have got to happen. Vaccinate those that are at highest risk. Don't vaccinate everybody else. Provide early medicines, whether they're repurposed or the latest thing from Pfizer or Merck. I really don't care, but right now the repurposed ones work pretty good as far as I'm concerned. Good enough that it would save at least 80% of the deaths.
And that's like ivermectin,
estimates.
chloroquine phosphate.
Ivermectin, hydroxychloroquine, vitamin D. That's the protocols we're going to work out in Rome in a couple of weeks, okay, at the International COVID Summit. And we're actually going to speak about those protocols to the Italian Senate in Rome. Boy, put that on your bucket list. Get to address the Senate in Rome. So that's
on the back.
So that'll come out in a couple of weeks. And I don't think I have to wear a toga, but I'm going to wear a white lab coat.
Yeah, why not?
So watch out for the knives in the back. So I've been told. So in any case, early treatment, existing drugs are pretty darn good. They're not perfect, but they're pretty darn good. Make available self-assessment tools, both for your risk for the disease and also whether or not you become infected and your risk for developing the more advanced disease. And we're going to have to kind of suck it up, put on your big boy pants. And this virus is going to move through the population, and that's just the way it is.
Right.
And
right.
And it will evolve, and hopefully it evolves to a less pathogenic, more highly infectious version, which is what is the usual course. Now I've got 6 minutes until the next one.
Okay.
And my wife has just bought me breakfast. So
I know. I'm so right. I'm so sorry. I just don't. I'd love to have you back. I don't know if I'm going to get you back. So I could ask the questions faster, but I guess one of the last.
No, seriously, I got to drop.
Okay.
We're done.
All right. Thank you so much for your time. Thank you so much for these answers. I do hope to speak to you again in the near future. Give your wife a big hug for me and everyone, and just have yourself a wonderful day. Take care.
Thanks. Ciao.
You've reached the end of our interview. Now, first, let me thank you for listening. I appreciate and respect you more than you'll ever know. And now I'd like to ask you a couple of questions. First, what 3 lessons did you just learn? What 3 aha moments just jumped out at you? Second, what can you implement for yourself and your business in the next 24 hours? Third, what can you give to someone else to help you with, or give to them to just do it for you? Whatever it is, remember, taking action is the secret sauce to results. Now, if you think this interview would be helpful for a friend, please give them a link to it. It'll help them and it'll help me too. I'd also like to invite you to help me find out more about the challenges you're facing, your dreams, your goals, and how I can help you overcome what's holding you back. We both do better when we know better, and your success is my success. So please reach out and interact. You can visit our website, bestbusinesscoach.ca for Canada or California, where I'm from and where I'm living. You're welcome to also try out one of our paid programs. You can find us on YouTube, Facebook, and pretty much every other social media channel you can think of. You should also subscribe to the podcast, and if you're enjoying them, please leave us a nice review. It really helps. That's all for now. Once again, thank you. Take care of yourself, and remember, the world needs the best business you can build. And I believe in you.